Hello, I am Anuradha Malik. In the previous episode of class 11 biology, we had learnt about the cell and cellular organization in prokaryotic and eukaryotic cells. Today we shall continue the lesson and learn more about the plasma membrane, cell wall and certain other organelles of the eukaryotic cell. First, the plasma membrane. The plasma membrane is composed of lipids arranged in a bilayer. The main lipid components are phospholipids, glycolipids and cholesterol, which is present in animal membranes. The lipids are arranged in such a manner that their polar heads are towards the outside and the hydrophobic tails are towards the inner part. This arrangement protects the non-polar tails from the aqueous environment inside and outside the cell. The cell membrane also possesses proteins and carbohydrates. The ratio of proteins and lipids varies in different cell types. Human erythrocytes have approximately 52% proteins and 40% lipids. Depending on the ease of extraction, membrane proteins can be grouped as integral or peripheral. The peripheral proteins lie on the surface, whereas integral proteins are partially or totally buried in the phospholipid bilayer. The widely accepted model of the plasma membrane is the fluid mosaic model, which was proposed by Singer and Nicholson in 1972. The quasi-fluid nature of the lipid layer allows lateral movement of proteins within it. This is why we call it fluid. The fluid nature of the membrane allows for cell growth, formation of intercellular junctions, excretion, endocytosis, cell division, etc. The membrane allows transport of molecules across it. It is selectively permeable to some molecules. Some molecules move across it without consuming any energy. This is called passive transport. A few ions are transported against the concentration gradient by utilizing energy. This is called active transport. Example, the sodium-potassium pump in the membranes. Water may also move across the membrane along its concentration gradient. That is, from the region of higher concentration of water to the region of lower concentration of water. Such diffusion of water across a semi-permeable membrane is called osmosis. Polar molecules cannot pass across the non-polar lipid bilayer. Transport of such polar molecules is facilitated by certain carrier proteins present in the membrane. The presence of a wide variety of proteins is the reason why we call this membrane a mosaic. Let's have a closer look at the structure of a plasma membrane. Outer to the plasma membrane is present the cell wall in plant and fungal cells. The cell wall is a rigid, non-living structure. The cell wall is present outside the plasma membrane. It is the cell wall which gives the shape to the cell and protects it from mechanical damage and infections. The plant cell wall is made up of cellulose, hemicellulose, pectins and proteins. Plant cells also have a middle lamella in between cells, which is mainly made up of calcium pectate. The middle lamella holds the neighboring cells together. The cell wall and middle lamella may be traversed by plasmodesmata. Plasmodesmata connect the cytoplasm of neighboring cells. The cell wall of a young plant cell is the primary wall. This is capable of growth, and as the cell matures, a secondary wall is formed on the inner side of the cell. Hemicellulose, pectin and lignin may also be deposited in addition to the cellulose fibrils in the secondary wall. Within the cytoplasm is present an extensive endomembrane system. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. A network of tiny tubular structures called endoplasmic reticulum is scattered in the cytoplasm as viewed through the electron microscope. The endoplasmic reticulum divides the intracellular space into two distinct compartments, the luminal inside the endoplasmic reticulum, and the extraluminal, that is the cytoplasm compartment. Often, ribosomes are attached to the outer surface of the endoplasmic reticulum. This now appears rough in appearance and is known as rough endoplasmic reticulum. Rough endoplasmic reticulum is seen in cells which actively synthesize and secrete proteins. The rough endoplasmic reticulum is continuous with the outer membrane of the nucleus. In the absence of ribosomes, the endoplasmic reticulum appears smooth and is called smooth endoplasmic reticulum. The smooth endoplasmic reticulum is the major site for synthesis of lipids. Let us look at a picture of the endoplasmic reticulum. Notice that it is in continuation with the nuclear envelope. Another component of the endomembrane system is the Golgi apparatus. Camillo Golgi in 1898 first observed densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. These are stacks of parallel, flat, disc-shaped sacs or cisternae of 0.5 to 1 micrometers in diameter. These sacs are concentrically arranged near the nucleus with a distinct convex cis or forming phase and a concave trans or maturing phase. Its main function is packaging materials for intracellular targets or for secretion outside the cell. Glycoproteins and glycolipids are found within these. Materials to be packaged in the form of vesicles from the endoplasmic reticulum fuse with the cis phase of the Golgi and move towards the maturing phase. Let us have a look at the picture of the Golgi apparatus. Notice the sac-like structures and the vesicles. Lysosomes are another component of the endomembrane system. These are membrane-bound vesicle structures formed by the process of packaging in the Golgi apparatus. They are bound by a single membrane. They are rich in hydrolytic enzymes which are capable of digesting carbohydrates, proteins, lipids and nucleic acids. They are optimally active at acidic pH. Because of the presence of hydrolytic enzymes, they are often also termed as suicide bags, because if they release their contents into the cytoplasm, it would digest the cell itself. The last of the endomembrane system, the vacuoles. These are membrane-bound spaces in cytoplasm which may contain materials such as water, sap, excretory products, etc. The membrane of the vacuole is called tonoplast. Contractile vacuoles, as in amoeba, are functioning for excretion. Food vacuoles are formed by phagocytosis, as in certain protists. In plants, certain ions are transported into the vacuole against the concentration gradient. Mitochondria. You already know that mitochondria are known as the powerhouses of the cell. These are called the powerhouses of the cell because the energy in the form of ATP is produced during aerobic respiration in them. Mitochondria can be visualized under the microscope with specific staining techniques. The number of mitochondria per cell, their shape and size varies. Typically, mitochondria are sausage shaped or cylindrical, measuring 0.2 to 1 microns in diameter and 1 to 4.1 microns in length. Mitochondria is a double membrane bound structure. The outer and the inner membrane divides its lumen into two aqueous compartments, the outer and the inner compartment. The inner compartment of the mitochondria is the matrix. The inner membrane forms a number of enfoldings called cristae towards the matrix. These increase the surface area. Specific enzymes associated with aerobic respiration are present on the two membranes and in the matrix. The matrix also contains single circular DNAs, a few RNAs and ribosomes of the 70S type. The mitochondria are thus semi-autonomous and divide by fission. Another organelle which is bound by a double membrane and found in plant cells is the plastid. The plastids are large and thus easily observed under the microscope. Based on the type of pigments, the plastids are of three kinds. First, the chloroplasts. These contain chlorophyll and carotenoid pigments. Chlorophyll imparts the green color and traps the light energy for photosynthesis during the light reaction of photosynthesis. Second, the chromoplasts. These impart yellow, orange or red color to the plant parts. They have pigments like carotene, xanthophylls, etc. The third are the leucoplasts. These are the colorless plastids with stored nutrients. Amongst these, amyloplasts store carbohydrates, elaioplasts store oils and fats, and eleuroplasts store proteins. Let us have a look at a sectional view of chloroplast. The chloroplasts are found in the mesophyll cells of leaves and other green parts of the plant. They may be lens-shaped, oval, spherical, discoid or ribbon-like. They vary in length from 5 to 10 micrometers and in width from 2 to 4 micrometers. The number of chloroplasts per cell may vary from 1, as in Chlamydomonas, and up to 20 to 40 per cell. They are bound by a double membrane. The space within the inner membrane is called the stroma. Flattened membranous sacs called thylakoids are present in the stroma. Thylakoids are arranged in stacks called grana. Flat membranous tubules called stroma lamellae connect the thylakoids of different grana. Thylakoids enclose a space called the lumen. The chlorophyll pigment is present in the thylakoid membrane, which is also the site for the light reaction of photosynthesis. The stroma of the chloroplast contains enzymes required for the dark reaction of the photosynthesis, that is, the production of carbohydrate molecules. Chloroplasts also contain small double-stranded DNA molecules and ribosomes of the 70S kind. Chloroplasts are also semi-autonomous and are formed by division of pre-existing chloroplasts. You must practice drawing diagrams of the sectional view of mitochondria and chloroplast. Today we have discussed about the plasma membrane, cell wall, the endomembrane system, the mitochondria and the chloroplast. In the next episode, we shall learn about some more cytoplasmic organelles of the eukaryotic cell.